Thank you again for that introduction. So my name is Ken Simon. I'm Senior Director of Protein Sciences and Biology at Revitope Oncology, where we're generating the next generation of T-cell engaging bispecific antibodies to revolutionize cancer immunotherapy to safely treat cancers and help patients live healthier and longer lives. And the title of my talk today is T-cell engaging antibody circuits, TEACs, that focus the immune activation to the cancer cell surface to widen the therapeutic index. And just to frame the discussion today, CD3 T-cell engaging antibodies or molecules, the landscape of which is really quite diverse. There are a wide number of targets where these molecules are being generated through a wide number of companies. But that said, there is only one approved drug, which is Blincyto or blinatumomab from Amgen, which is a CD3, CD19 bispecific T-cell engager. This is an exceedingly potent molecule, but it carries with it fairly significant toxicity issues, in addition to what I would consider poor drug-like properties or a very, very short half-life. And this is really the hallmark of T-cell engaging therapeutics, or at least the first generation of such therapies. These molecules, are highlighted on the left. They're very toxic. They're very potent at targeting both tumors and tumor metastases. And they do this through immune activation, but this immune activation is poorly targeted, and it results in widespread T-cell activation, triggering cytokine release syndrome, and what are routinely fairly severe adverse events. What we do at Revitope is take this technology and engineer it so that it's a precision therapy where we target the tumor, we activate the immune system, but that immune system is delivered specifically to the tumor to avoid systemic toxicity. Our molecules are modular by nature. They're generated from components of primarily antibodies. And these are antibodies where, for example, the CD3 engaging domain is coming from a clinically proven T-cell engager. The targeting domains are frequently derived from antibodies that are being utilized clinically. And then we have what we typically refer to as the dummy domain. It's a masking domain for the CD3 engager that also acts as a molecular stabilizer. And then finally, we have a half-life extension module that gives our molecules a significantly longer half-life than a traditional bispecific T-cell engager. The mechanism of action of these molecules is a three-step process where our two half molecules, so our system is designed as two half molecules, each target a unique cancer antigen. So in step one, our molecules simply bind to the antigens on cancer cells. In step two, you'll note that there is a proteolytic cleavage site separating our dummy domain and half-life extension domain from the CD3 domain. So these are proteases that are selected as being enriched in the tumor microenvironment. These proteases cleave the linker within our molecule, liberating the dummy domain and the half-life extension domain, leaving a antigen binding domain with a CD3 half paratope. In the context of a cell surface, these half paratopes will come together, they dimerize, and in this conformation, they can now engage CD3 in the surface of a T cell, bringing that T cell in close proximity to the cancer cell and driving the elimination of the cancer cell through this artificially driven T cell activation system. So our approach to our system is selection of the antigen binding domains. We typically go after the low-hanging fruit. These are antigens where there are known antibodies with clinical experience and freedom to operate. We perform initial expression analysis because it's fast and easy. Once we select our antigens of interest, we do an extensive biology review to ensure that these are going to be, are likely to be cancer specific, not widely expressed as on the cell surface of other normal cells together, which is an important point. And then finally, we move on to extensive protein expression analysis through a combination of immunohistochemistry and proteomics, where we identify what we will then select as a good antigen pair. So this is our screening funnel for selection of our antigen binding domains. And the precision of our system comes from the fact that on a normal healthy cell, you can have an antigen A expressed, and our molecules will target that cell. Typically, we would expect limited proteolytic activity within normal tissue. So our linker is not cleaved in this situation, and we will have a binding event of an inert, what is effectively a prodrug. The same thing can happen with antigen B on a different cell. And even if there is protease activity within these cells, we generate a half paratope for CD3 that would still be inactive. But in the cancer situation, where you have both of these antigens together, you get binding of both molecules, complementation of the CD3 half paratopes, which will drive T cell activation and promote killing of the tumor cell by the T cell. So next, so our components, the critical aspect of this design is the CD3 module. We've selected an anti-CD3 antibody with clinical experience. Our molecule crosses to cynomolgus monkeys, so it makes development of our candidate molecules easier. We have freedom to operate. And then we've taken these molecules and done structure-guided mutagenesis to engineer out the activity of the half domains, creating these dummy molecules. So for the light chain, we have a single amino acid substitution in the light chain dummy and 2 amino acid substitutions in the heavy chain dummy. And both of these molecules are now inactive, and their half paratopes do not bind to CD3. And I'm going to show that data right now. So in this experiment, it's a little bit complex of a slide, excuse me. But if we focus on the left, this is simply a CD3 ELISA. And we are comparing in this slide a bispecific T cell engager molecule. So that is shown here. It's got an anti-CD3 domain. It's got an anti-antigen domain, but this is an antigen-independent experiment at this point, where we've put CD3 on a plate. The bispecific T cell engager can bind to CD3, and then we detect the BiTE molecule. If we take either the our TEAC molecule, the VH half or the VL half that are shown here, these are cleaved by recombinant protease molecules. So our masking domain has been eliminated. Neither of these molecules, either the VH or the VL half of these molecules, can bind to CD3 alone. Additionally, if we mix these molecules together, so we have both the VH and the VL that are demasked, the affinity of the VH and the VL in solution is quite low, and it's insufficient to complement our TEAC molecules in solution. Now, if we move to the right side of the slide, We do the same experiment, but here we're putting the tumor antigen on the plate. In this case, our antigen binding domain can bind to the tumor antigen. It's holding the CD3 domains in close proximity, so it's therefore effectively increasing the local concentration of our half paratopes, mimicking what we would expect to see on a tumor cell surface. And in this conformation, our paratopes come together and we can bind to soluble CD3 in this case, and we can detect that. And so what we show on the graph here is that the half TEAC alone, either the VH or VL, have no signal in this sandwich ELISA. If we have removed the mask from our TEAC molecules, we get a significant binding event for CD3, but in the mask conformation, again, these molecules do not bind, demonstrating that we can reconstitute our CD3 binding domain in the context of a two-dimensional surface, but not in the context of a three-dimensional solution phase. So, and how about the protease activity? So this was assessed, and this is the only bit of protease that I'm going to talk about in this presentation today, but just note that we spend an extensive amount of time focusing on the selection of our protease cleavage sites and the protease activity in tumor cells. But we have a standard site that we use, which is MMP 2 and 9. And that's pretty sufficient for most of the experiments that we do. So in this case, we're looking both in vitro and in vivo in tumor-bearing animals, where we either incubate our TEAC molecules on cells, and these are cells that are expressing an antigen that is specifically bound by our TEAC molecules, or we implant tumors in mice, and these tumors again have the antigen that is specific for our TEACs that we've used in these experiments. Either the cells or the tumor are harvested from these animals. They're lysed. The TEAC molecule is immunoprecipitated, and we run that material out on a Western blot, and we can measure both the intact versus cleaved molecule. And looking in vitro, you can see in the Western blot at the top or graphed on the bottom that in a time-dependent fashion, our molecules undergo proteolytic cleavage, and you see an increase in the cleaved molecule and a concomitant decrease in the intact molecule. And by mass spec, we've analyzed these molecules, and these molecules are in fact cleaved at the expected protease cleavage site. We do the same thing in vivo. We collect the tumors. It's a little bit more of a dirty experiment due to the in vivo nature. But again, we see a time-dependent increase in cleavage of the molecule and reduction in the intact molecule within the tumor of those animals. Importantly, we also look in serum. So in this case, we've injected animals with our TEAC molecules. This is a non-tumor bearing animal. That red dot is a misnomer here. Plasma is collected. The TEAC molecules are immunoprecipitated from the plasma of these animals. We perform our Western blot, and you can show that over time, there with a little bit of variation, there is effectively very limited cleavage of our molecule in serum over time, and the intact molecule remains stable. So our molecules are cleaved within the tumor environment, but they are not generally cleaved within the serum compartment of these animals. Next, we wanted to determine exactly how much of this protease activity do we need in order to achieve efficacy of our molecules. So this is a little bit complex of a slide, and it includes some experimental data and some systems biology modeling of our data. On the far left, we're using interferon gamma release from T cells and cancer cells that are treated with our TEAC molecules that have been pre-cleaved and mixed such that we have 0% cleaved molecule, 2.5% cleaved molecule, 10% cleaved molecule, 30%, or 100% cleaved molecule. We do a dose response on cells looking for interferon gamma release as a surrogate for T cell activation. And then we generate the following curves. We then take the EC50s from each of these curves, plot that by percent cleavage of our molecule, and we show that going a little bit backwards, but going from 100% cleaved to 30% cleaved to 10% cleaved, the EC50 is very low in each of those cases. And after, when you get below 10%, the EC50 of T cell activation has a very rapid rise, suggesting that, and we drew this line at 20%, where we suggest that if you get 20% cleavage of the molecule, and by the way, this is a PC3 cell system. So this is one antigen binding domain, one cell type. So we're going to generalize and we're going to test this in the future, and including experiments that we've already done. But what we argue is that if you achieve 20% cleavage in the tumor cell, that any additional cleavage beyond that, there's very little impact on potency. And less than 20% will result in a big impact on potency. And again, I reiterate that this is a single cell system. We then took this data, and in collaboration with Applied Biomath, they generated models based on this data, looking at the expected cell killing versus the cleaved TEACs. And they did this in the context of varying the antigen density on the different cells. And by doing this, they arrived at this 20% cutoff molecule. And actually this 20% preceded our drawing the line here for these molecules. So this data suggests that if we achieve 20% cleavage of our TEAC molecule, that we will have sufficient in vivo potency, and anything less than that, we would expect our potency to drop. Or in the context of normal tissue, if we have less than 20% cleavage, should there be dual antigen binding, we would expect little toxicity in that situation. So we then moved on to look at a comparison of our molecules to bispecific T cell engagers. And we looked at T cell activation by the induction of a variety of cytokines. So in this case, we used a bispecific T cell engager with antigen A versus a combination of VH and VL TEAC molecules against antigen A. And I should point out that when we build our TEAC molecules, we can actually build them against a single antigen. We just have to build the two molecules, the VH half of that molecule and the VL part of that molecule, or we can target two independent antigens. For the sake of these experiments, or this particular experiment, we're looking at a single antigen TEAC. So it's the VH and the VL version, but the antigen binding domain is against a single target. In each of these cases, we show similar induction of interferon gamma, IL-2, IL-4, TNF alpha, IL-10, and IL-6 with our VH and VL TEAC as compared to what one would see with a bispecific T cell engager. And then this black dot on the bottom right of each of these is representative of our control, which is the VH or VL TEAC molecule alone in the absence of its partner. We then look to see whether our TEAC molecules can be cleaved in a variety of cell lines. And now we're just looking at interferon gamma as a surrogate for T cell activation, but we can show that in two different prostate carcinoma cell lines, we see induction of interferon gamma, as we also see in lung tumors, colorectal cancer, as well as breast cancer. So this is a generalizable activity that we see across multiple tumor types. And in collaboration with an unnamed large pharmaceutical company, we had them explore our molecules on two different antigens. So antigen A is expressed at 30,000 copies per cell. Antigen B is 32,000 copies per cell. And looking at three different T cell donors. In these experiments, we're looking at both HCT15 cells, which is a colorectal cancer line, as well as the MDA MB231 breast cancer cell line. In this case, we're looking at antigen A, and in this case, we're looking at antigen B, and we're comparing the bispecific T cell engager shown in blue versus the TEAC molecule, the VH-VL combination TEAC molecule for antigen A at the top and antigen B at the bottom. And then there's a whole grouping of controls of the tumor cells alone, PBMCs plus tumor cells in the absence of molecule, or the half TEACs alone. And we show that our molecules are very comparable to the activity across donors in T cell mediated killing of these cells as compared to the bispecific T cell engagers. Our TEAC molecules are very sensitive, though, to the expression level of the antigen on these cells. So in this example, with antigen A and B expressed at roughly 30,000 cells, we also did experiments where the expression level varies from cell type to cell type. So for antigen A on MCF7 cells, which is a very high expression level, our molecule behaves very similar to that of a BiTE. And you can see the EC50 shown by the red arrow here. In a PC3 prostate cell, the expression level is medium. You see a rightward shift in the EC50 of the BiTE molecule, of the TEAC molecules, whereas the BiTE molecule remains stable. And then looking at MDA MB231 breast cancer cell lines with a very low expression of antigen A, our TEAC molecules failed to induce interferon gamma in that experiment. And so the top panel is interferon gamma release. In the bottom panel is specific T cell mediated cell lysis. So our molecules, the activity of our molecules does correlate with antigen expression. And that's a distinguishing factor from the bispecific T cell engager, where if you have low level expression in normal tissue, the BiTE molecule is going to induce toxicity within that tissue, whereas the TEAC molecule would be expected to not have that effect. In this experiment, we show the difference between a TEAC molecule targeting antigen A alone, shown in red here. So this is a VH version and a VL version with antigen A binding. And we're comparing that to a dual targeted TEAC molecule where we are binding to both antigen B and to antigen A, each with a VH or VL molecule, showing that both T cell activation by interferon gamma or T cell mediated killing are very similar in these PC3 human prostate cancer cell lines. In addition to this, another feature of our system is that a high expressing antigen can rescue the activity of an impaired low expressing antigen. So in this experiment on MB231 cells, antigen A is not expressed at sufficient levels to induce specific T cell mediated cell killing. So this molecule, up to 100 nanograms or 100 nanomolar, has very limited activity, whereas the BITE molecule of antigen A has very high activity because it's less sensitive to low expression of target. If we then bring in a dual TEAC with antigen B, where antigen B is expressed at moderate density, we can then combine that with antigen A and rescue this lack of activity by incorporating a more higher expressed antigen on the surface of those cells. The importance of this point is that if we can find antigen pairs where one of the antigens has less tumor specificity than the other, but is higher expressed, we can combine that with a highly specific tumor antigen that's expressed at a level that's low enough where we wouldn't expect TEAC activity. Therefore, we can bring the activity up of a low expressed antigen with a TEAC in a way that would still provide that combinatorial selectivity and reduced expected toxicity in man. And finally, this is a very important point. These molecules are, in fact, very complex molecules. And the orientation of our molecules matters. And not every antibody that we use in a TEAC actually makes a good TEAC. So a molecule that's a great monoclonal antibody against a target may actually not be a very good TEAC. And we can use that in some cases to dial in the activity that we like. And we do this by switching the orientation of the VH and the VL. So in this experiment, we have a BITE molecule. It's very potent. We have a a single antigen TEAC, antigen A, VH, and VL, as shown by the blue squares, which is very potent. If we combine antigen A with antigen B in the antigen A with a VH and antigen B with the VL, our activity is not as potent as antigen A alone, and this would be expected because antigen A is not expressed as highly as antigen B is. If we switch that order and we put the VH on antigen B and the VL on antigen A, we see a decrease in activity. And that suggests that the geometry of binding of these molecules is actually a critical component. So when we build our molecules, we actually will select a number of antibody domains. And we build these in both the VH and the VL formats. We build them in both orientations. And we are selecting our molecules to maximize the potency in order to, to derive our molecules with really the best drug-like properties that are the most potent that we can achieve. Okay, and then finally, this is really preliminary data that we've done in-house, but our data suggests that our TEAC molecules are stable and have reasonable drug-like properties. So we've done comparing to a fresh thaw of this molecule. In this experiment, we've done 2 freeze-thaw cycles. We've actually extended out to well over 10 freeze-thaw cycles, and we see really no difference in activity. So this is a bit of an old slide. We've compared 7 days at 4 degrees to 220 degrees at 4 degrees. And actually that number is significantly longer than that at this point. And we show very little change in activity of these molecules. In addition to this data, which I'm not showing today, we've done stability studies at elevated temperatures, and we currently have an experiment through a contract, a CDMO organization who's doing bona fide developability studies with our molecules. And those molecules, those studies are in process currently. And and are currently looking like our molecules are actually behaving very similar to what you would expect for a monoclonal antibody. And then finally, we have our half-life extension domain, and we're comparing in this experiment. So this is cell killing on the left and interferon release on the right, compared to a BITE molecule for an antigen A, half-life extended TEAC molecule, so it's got this extra half-life extension domain, versus a molecule that lacks this domain. And you can see that the red and the green lines are falling right on top of one another, that this half-life extension domain is not impacting in any way the activity of our molecule with respect to T-cell activation, but showing on the right, this is a single dose PK study in non-tumor bearing animals comparing the half-life extended molecule shown in the purple line versus the non-half-life extended molecule in blue, with a half-life extending from 6 hours to roughly 43 hours in this experiment. So that's, this is our final format of our molecule. And we next moved our studies in vivo. So taking MB231 breast cancer line into NSG mice that are implanted with MB231 cells grown to about 75 nanometers, cubic millimeters. These animals are engrafted with human PBMCs, and they're dosed with the BITE molecule every other day at 100 micrograms per dose, or TEAC molecules at either a drug total of 260, 26, or 2.6 micrograms once per week. And although the lowest dose had no statistically significant effect on the tumors in these animals, at the 26 and 260 microgram per week doses, we saw a statistically significant inhibition or regression of tumor growth in these animals. And the BITE molecule knocked that tumor volume down to 0 in all animals. So in this study, it's a single TEAC antigen. We repeated that experiment using dual antigen, where in this experiment, the dosing was 200 micrograms total dose of TEAC molecule for the antigen A single antigen TEAC, which was ineffective in this HCT15 colorectal cancer line. Antigen B completely as a TEAC molecule or as the BITE molecule, completely obliterated the tumors in this study. Now when we combine antigen A and B, we again can reconstitute or capture the activity of antigen A and bring that activity down to what now is an efficacious molecule, demonstrating that our dual targeting of molecules will target tumors in vivo. So our approach in both can be applied to both solid and hematological tumors. In solid tumors, there's a high unmet need. We have great dual targeting for excellent differentiation of our molecules versus our competitors. There's many reports of abundant protease activity in the tumor microenvironment in solid tumors. But that said, there's limited evidence for T-cell redirection in tumors, and we hope to change that situation. For hematological tumors, there is experience with T-cell redirection. We are exploring those ideas, but there's much limited protease expression in those tumors. So I'm just going to summarize by saying that Revitope's dual targeting two-component TEACs offer a unique approach with 3 specific control mechanisms. It's the protease activity, it's the antigen binding, and it's T-cell redirection. We have elegant protein engineering for a modular adaptable framework. We've demonstrated our molecules are effective in vitro and in multiple in vivo models. I've only shown 2, but we've done many more than that. And this is a promising treatment for both solid and hematological tumors. And I'd finally like to acknowledge our Revitope team, our partners at Applied Biomath, our founders, which are derived was coming out of Mark Cobalt's lab at Mass General Hospital, and then finally down here our group of consultants for complex areas of biology. And I'll finish with stating that Revitope is currently hiring, so please check our website for if you know of anybody or if you're interested in exploring career opportunities with us. And I will finish with that.
Wonderful. Thank you so much, Ken, for your presentation. And yeah, as you mentioned, there are positions open in Revitope, and if you have a CV that you want to forward, do let us know as well, and we'll go forward there. And there are a couple questions for you. One is from the Chief Scientific Officer, Dr. Jim Keith from Amstel, PA. He said, "Very nice work, Ken. Have you done any cost of goods analysis for the production of your TEAC molecules? These are complex biomolecules, so it would be important to know how they express and what the cost and supply of commercial product projections show."
So we've not done specifically actual cost of goods analysis. The expression is similar to monoclonal antibody. That said, it's a two-component system. So initial CMC costs are expected to be double, they're expected to be double what a single molecule might be. Our plan for fill finish in the same vial of our 2.5 molecules is something that we would discuss with the regulatory agencies to try to bring down the CMC costs. And we do have some concept molecules where we might be able to build our system as a single molecule that would then bring it back towards a what one might expect from a single agent. With respect to expression, our expression levels are not significantly different from that of a monoclonal antibody.
And there's another question for you from Mark, head of biology. He's asking, in preclinical models, do you see emergence of resistance due to loss silencing of one targeting antigen?
This is something that we think about quite a bit. So when we select our monoclonal antibodies as our targeting antigen, one of the things that we screen out is downregulation of the target or internalization of the target. We want our target to remain on the cell surface so we can reconstitute that CD3 domain. But that said, if there is downregulation of our target, there could be resistance under those situations. And that resistance would be similar to what one would expect with a bispecific T-cell engager or a monoclonal antibody against the same target. But it remains a theoretical issue.
And there's one last question for you from Larry Hardy. He said, very innovative engineering, a possible concern. Do you anticipate inter-individual variability of tumor antigen expression or cleavage protease could lower the efficiency toxicity ratio for your chimeric molecules in some patients? If so, how could you mitigate such problems?
We do extensive studies on the expression of our antigens of choice through validated IHC assays through a CRO. And as we nominate our candidates moving forward, we collect increasing numbers of patient-derived tissue samples to actually screen for the expression of our antigens. And this is something that we consider. It's also an assay that could be used to support clinical studies for patient selection as for candidates. With respect to the protease, I didn't really talk about that. We have a very, very large program where we're exploring the heterogeneity of protease activity within the tumor microenvironment, and the selection of the proteolytic cleavage sites within our molecules that would be best for any given tumor indication.
Wonderful. And yep, that was Ken Simon. And like you mentioned, if you or your network is interested in applying for these open positions, let us know or reach out to him directly. Once again, Ken, thank you for your presentation.
Thank you for having me. I appreciate it.
You're welcome.